they have an enzyme for H's. We learned that RNA is required, DNA is required to make RNA, and RNA is required to make protein. And that is true in most cases. That's how life proceeds, basically. However, when we have, and this was discovered in viruses, when we have reverse transcriptase, this enzyme is actually able to go from RNA to DNA. So that's exactly what these viruses do. They can use this positive or sense RNA to make a copy, not a copy, to synthesize DNA. And also keep this in mind. Viruses need DNA to replicate. And they need RNA to make proteins. And this is important because of the capsid. Okay? If you have a virus that's this is the capsid and this is the DNA. So they need replication and they need translation for those two different components. So that's why we are dealing with some viruses that have to go through DNA intermediates before they go to RNA particles. And group number 7, which is double-stranded DNA, that they need also, or they use a reverse transcriptase to make double-stranded DNA. So now let's make a stop and look at these figure questions, because this is extremely important that you get all these different criteria that we use to classify viruses according to this particular classification system. There are other molecules, you have to be aware of these differences. Here we have RNA, and here we have mRNA. Anybody knows the difference between these two?
The introns, it's already been compiled, right?
In which case?
In mRNA. So the exon's been taken out.
Yeah. What's another way to put that? That's true. So this, when we say RNA, we are referring to the whole. Remember that RNA exists in 3 different particles. Ribosomal RNA, transfer RNA, and mRNA. But at the end, for translation purposes or to form proteins, the only one that matters is mRNA, because that's the molecule that carries the information onto the ribosomes for protein synthesis. So that's why here we have, this is just the general structure of the virus. And if it's anything like bacteria, of this, if this is 100% of the RNA in the virus, about, at least in bacteria, this mRNA represents barely 3 or 5% of the total RNA. Okay. Now in viruses, that number, I'm not sure what it is. It might be different, but only a portion of this RNA ends up being mRNA. So that's another difference that you have to keep in mind. And we have, again, double-stranded DNA and single-stranded DNA. Remember, for viruses to replicate, to make copies of themselves, they have to have double-stranded DNA. So the RNA viruses and the single-stranded DNA viruses, they have to have this intermediate stage. So these are just some notes. Group 1 are the normal, consider the normal double-stranded DNA that are transcribed directly into mRNA and it replicates inside the host nucleus using the host's DNA polymerases. Group number 4, the genome is positive sense RNA, and they can be actually used directly as a template to make proteins. And 6 is sense RNA that can be used to form mRNA but they also have this reverse transcriptase enzyme to go from a single-stranded RNA to double-stranded DNA. And that's why they call retroviruses. And the most common example that you'll find is the HIV. Actually, some of the not vaccines. Some of the therapies used to treat HIV is to block the function of the reverse transcriptase. If you block that, you make it impossible for the virus to make copies of itself. And it has to make copies of itself in order to survive. So now that we know more about the classification system, we have to see how they actually replicate. And these are the 4 general stages of replication. First, they have to recognize the host and then attach. They have to insert the genome into the cell. They have to replicate the genome and then assemble the virions. And then they have to exit the cell and infect another cell or more cells. And have this. This is not on your handout. I can pull it up later because I just found it useful to explain the different processes. We have a virus that's in the outside. So the first thing that this virus has to do is recognize a cell that it can actually infect. Okay. If this is, let's say this is HIV. If this was a plant cell, this cell, or not even a plant cell, a non-human cell, non-primate cell, this virus would not be able to recognize the cell. But it does, or let's say it does. So it recognizes the cell so that it attaches to the cell. In this case, what happens is that the cell will, because it doesn't recognize the virus as a foreign particle, it will absorb it. In the membrane of the cell will absorb the viral particle. Then the viral particle will penetrate the cell. Now we have this naked capsid, basically. Once this happens, the virus has to be uncoated. It loses the capsid of the cell, of the virus. Then once it's naked inside the cytoplasm, it will replicate in the nucleus of the cell. Once it replicates itself, the ribosomes of the cell will produce these proteins that form the capsid, and they will assemble the viral particles, and then they will be released back to the outside. So it seems like a very simple life cycle. And it is simple, it's just that different viruses will have slightly different strategies to follow these 6 different stages. Questions on this life cycle? Anybody knows what are the molecules that are the receptors on the cell surface for viral recognition? What kind of molecules do the virus actually recognize in the cell so they can attach? They are either glycoproteins or glycolipids. And these receptors are in the cell because the cell uses them for things like nutrient absorption. So it just happens that those same molecules are used by the virus to attach to the cell. Okay. So using bacteriophages as an example, this is basically what happens. Bacterial cells receptors are normally used for functions like the uptake of sugars. So this is a T4. It attaches to a bacterium. Once it attaches and it's anchored by these other structures, then the DNA is injected into the cytoplasm. Now there's a difference between, if you see the difference on how the DNA enters the cell between these particular T4 and these HIV viruses. So that tells us the different viruses will have different mechanisms to inject the DNA into the cells. Here, basically, the whole particle is absorbed into the cell, the whole cell, whereas at before, it doesn't need to. The capsid never actually enters the cell. It just has this mechanism to inject the DNA. So after this DNA transfer happens, this is empty, and it is basically a ghost, a capsid. It detaches from the cell, and it goes away. I mean, it doesn't go away. It probably degraded. Now, when it comes to all viruses, they undergo different cycles. They can undergo two different cycles: lytic or lysogenic cycles. The main difference is that during the lytic cycle, the bacteriophage will replicate very quickly, will make copies of itself, it will kill the host cell, and it will release the particles to the outside to infect new cells. That's the easiest way to replicate and become infectious. Or they can become lysogenic. Lysogenic implies that the bacteriophage basically integrates its own genes into the cell chromosome, and it will remain there in a dormant state. So every time the cell replicates, it's replicating the virus genes as well. And it can be in that particular stage indefinitely, until it reactivates and then becomes lytic. You see? In the lytic cycles, this replication happens quickly. In the lysogenic, it remains dormant. So these are called temperate viruses. And what makes the lysogenic cycle of viruses become lytic? Basically the environment. When cells become stressed, there are mechanisms that the cell has to perceive that it might die, basically. So if there is any environmental stress, then the bacteriophages that are included in the chromosome, or the prophages, will undergo lytic cycle to make copies of themselves and infect new cells before the cell dies. Because if the cell dies, the virus will die. That's another reason why, when I was talking about, like in this case, if this was a dead cell, this interaction wouldn't happen. So that's why viruses do not infect dead cells. Okay, that's when in the lab you want to culture viruses, you have to grow this viruses in cell cultures. And again, this is a central figure to understand the two different life cycles. Here we have the host genome. This is the bacteria. This is the genome, or the chromosome. And we have a phage particle. The phage particle attaches to the cell and injects its nucleic acid, in this case DNA. Now, this DNA, if you notice, it has to recircularize first before it can be recognized by the cell ribosomes. So now this genome can be copied or replicated and packaged, and it makes, once it has different copies of itself, it bursts the cell and it releases these new phages to the environment. This is the fastest way to replicate. Or, if it's lysogenic, this genome will integrate itself onto the host chromosome, becoming a prophage. Now, every time this bacteria or cell replicates, it will also replicate the viral genome. The integrated phage DNA reproduces with the host genome, so you have different generations, and all the generations will include these prophageous genes as well. And it can remain in this lysogenic state for, not forever, but for many generations. Our genome has genes that are biologic origin, that we don't know yet how we got them, or when did we get them, or what are they for. In some cases, these insertions give the bacteria characteristics that they wouldn't have otherwise. I think in the case of Ebola, no, not Ebola, this very infectious disease from water, contaminated water, I don't remember the name. But in some cases, the bacterial outbreaks are basically due to viruses that have entered the cell and made it virulent. So virulence factors, in many cases, come from viruses. And I guess that's where the name comes from. So once we have this prophage in the host genome, if these cells are under stress, that will induce the excision of the phage DNA. You see how here is basically breaking apart or coming from the genome. And once it enters this stage, it can become lytic. So some genomes and some viruses will be lytic, and some will be lysogenic, but at the end, lysogenic viruses end up being lytic at some point. Questions?